Thanks, everyone. This is kind of funny. It's like we should just kind of all sit together. But yeah, so I recently moved to Temple to join the MDA ALS Center of Hope and the ALS Hope Foundation. I still live, I came from Hopkins, and I still live in Baltimore. My wife still works at Hopkins, too. I'm on service right now, literally in the hospital. And some of you know Dr. Hyman Patterson, I think, who has like infinite energy and puts us all to shame every day, and is the reason that I came to Temple. And so she's covering rounds for me right now. So if my pocket dings, like if I ignore her, I'm in big trouble. But anyway, so yeah, we're gonna talk about ALS research and kind of an update on the sorts of things that are going on. And as far as my disclosures go, most of the stuff listed there is actually stuff that I'm a volunteer for, but it still is a disclosure. And for example, I chair the programmatic panel of the Department of Defense ALS Research Program, which is right now the largest dedicated funder of early therapeutic development, early testing in ALS, and actually Dr. Harms serves on the panel with me. But that's a volunteer job, and so I don't represent them. I'm not here talking on their behalf. I don't represent, I'm on the board of a couple foundations, and I don't represent them. For me, it's a privilege and an honor to get to do this stuff, and I'm going to talk a little bit about why, and most of it is interacting with you guys, and ALS warriors, people with ALS, caregivers, advocates, and you're my Kool-Aid, so I work for you, and I'm happy to do anything that I can. So this is Jack Griffin. Dr. Griffin was the chair of neurology at Hopkins when I came actually as a visiting med student and then was a resident. I inherited his clinic, in fact, when I joined the faculty. He's now passed away, but he was also the editor of Nature Clinical Practice Neurology, and he used to write these editorials. And this is one that he wrote, and I want you to notice this is written in 2006, right? And so the title is Research in Neurological Diseases, Lost in Translation. The brain sciences are on a roll. Young scientists are choosing the neurosciences for their careers. Basic discoveries are accruing at a breathtaking rate, and yet the relevance of these discoveries to neurologic disease, is increasing. But then look at the part in yellow there. Nevertheless, there's a palpable restlessness among disease advocacy groups, philanthropists, and neurologic investigators. And why? Because treatments for these diseases simply are not emerging at the rate that we might have predicted. And again, this was written in 2006, but up until maybe a year ago, I kind of feel like I could have written this right now, right? And so very recently things are changing, and very recently we're starting to get some signals and some drugs, but what's the reason for this, and how can we do better? And so, this editorial that he wrote, it concluded with some suggestions, and again, back in 2006, and it talked about focusing on speed and setting aside flexible funds that can be directed promptly and in the unfettered exchange of ideas and data, and developing new IP approaches that don't delay progress. And this is the stuff we still talk about today. It sounds futuristic, and yet it was written quite some time ago. We finally are making some progress, though. We finally are figuring out how to all be in the sandbox together, how to work together, how to collaborate in ways that matter and accelerate this stuff, and it's exciting. And we're going to talk about that. So this slide has two quotes from two different people who are two of the amazing people that I get to interact with, right? So some of you might know Dr. Scheffner. Dr. Scheffner is an ALS clinician, a researcher. He runs trials. He's at the Barrow Institute. He's involved with lots of different efforts in ALS. This is a slide I stole from him from one of the clinical research learning institutes, right? So these are these things that are run by the Northeast ALS Consortium to train people living with ALS and their caregivers to be research ambassadors, to go out in their communities and talk about ALS and get people involved in science. And so this was a talk that he was giving about how can we do better making drugs. And he says, what makes a successful trial? So you need good preclinical science, you need adequate pharmacokinetic information, so that's understanding how the drug changes after you put it in the body and how long it sticks around. You need something you can measure, right? A lot of the problem with ALS is that we don't always have things that we can measure that are sensitive, that we can tell if somebody's changing. And you need to make sure that the trials are well designed. And then on the right side of that slide is actually a Twitter feed from Nadia Sethi, who is also a really amazing person who I get to work with. She's actually on the programmatic panel for the CDMRP with Dr. Harms and me. She's what's called a consumer member, meaning that she's not an ALS physician or researcher, But that's part of the amazing thing about that program is we're all doing this together. And so she says, drug development is difficult. Drug development for ALS is even more difficult. Doesn't mean we won't succeed, does mean we need trials now, patient-centric trials. We're going to talk about what that means a little bit. But there should be a damn good reason for a drug to be in a trial. Strong preclinical science is crucial. And you know, a lot of times the questions that we hear from people living with this disease to us is, why aren't you guys trying this? Why is this in a trial? I saw this thing, it sounds like an awesome idea, why can't we do that? You know, and I want to talk about that a little bit. How do things get to trial? Who chooses, right? And why? And what does it mean to have strong preclinical science? So, you know, you often hear people talk about a research pipeline, right? Like this is we have things in the pipeline of our company and we're developing therapies. And it's always kind of struck me as a weird description, a weird word for what we're doing. So, you know, what a pipeline is a continuous tube, right? Something goes in one side and then there's no interaction with its environment at all until it comes out the other side. And maybe once upon a time that's the way research was done. Maybe once upon a time somebody did something in a lab and they got a result and they handed that off to the clinician and then they were not involved anymore. And the community wasn't involved, and the advocates weren't involved, and the MD wasn't involved, and it just went out the other side, and you did your trial. But it's really not representative of what we do now. The other thing that a pipeline is like that surfing pipeline. I really don't like that metaphor. Like, forget that. Bottom right there, I have a 19-year-old kid. He played Pipeline Soccer Club was the name of his soccer club, and I think it's because they think it's rapidly, I don't know, bringing them to a result. Who knows? You know, again, this is not the way we operate today. And when I think about, so Dr. Griffin wrote that essay in 2006, right? And you know, if somebody gives a research update talk in ALS, like every year they're going to tell you, ooh, I'm excited about this and this gene therapy, and you know, it was some other therapy a few years earlier. If I really step back and think about what's changed, it's the way that we are all working together. It's truly, and by all of us, I don't mean scientists and clinicians, I mean advocates and people with the disease and caregivers and anybody and everywhere is really sharing the work and helping each other, and that's really not represented by a pipeline. In fact, a pipeline is kind of an eyesore on the landscape, right? So, you know, but this is the kind of slide that people show when they talk about drug development pipeline. And so you've got early studies looking at risks and you know, what might cause somebody to be at risk for a disease. In ALS land, you know, a lot of that is done by the CDC, right? In the CDC National ALS Registry. And then, you know, basic science, trying to figure out the biology of the disease, trying to figure out what are models that we can use to test things in the lab. And, you know, certainly there's a lot of funders in this area. MDA is one of them. And NIH, I think traditionally is maybe who we think of in this country as the bigger part of this. The Department of Defense program that I mentioned that we are volunteers for is sort of in this space here. So it's not pure basic science, but taking that basic science through to a therapeutic, to an early stage trial. Whoops. And so the idea is that at the end of that, we hope to have things that are, for example, ready for the Healy, you know, the platform trial, right? With good, rigorous data, and we can have, you know, successful trials, hopefully. My dad's a doctor, and when I was a kid, when I was growing up, he had this hanging on the wall in his office, and it really, like, I'd never understood it. I'm not quite sure I still do. I think it's probably a New Yorker cartoon, right? It says, you know, when did I decide to become a cowboy? I thought we were doctors, and like, not only did I not think it was funny, but I thought it was dumb, right? Because of course these aren't doctors, they're cowboys. Look at their hats, right? That's a cowboy hat. And when you're a kid, like every job you think of as a kid, like most of them have cool hats, you know, like football player or, I don't know, fireman, airplane, you know, pilots. Doctors really don't have cool hats. But anyway, I thought of it recently, and I thought of it in a slightly different context, and that's this context that these people that I get to work with, and some of them are like Dr. Harms and Dr. Hyman Patterson, but most of them are Nadia Sethi, and most of them are people like you guys who, it's just ridiculous how many different things you do in the ALS community and serve so many different roles. And we talk about somebody wearing many hats. And so I was insomnia surfing in the middle of the night. I'm like, where does that come from, like wearing many hats? And to be honest with you, the internet's not sure, and there's a couple theories. And so one of them is that there's this play from the 1800s by Gilbert and Sullivan called The Mikado, and there's this character in it, he's called the Grand Poobah, right? And in the town of Titipu, he basically holds, I think there's one guy who held one other job, but he held every other job. And part of the amusing thing about the play is he like runs around from job to job, and the way you know what he's doing is he like switches hats, right? So he's like in the bank wearing his bank hat, and then he runs off and he wears the other hat, and it's supposed to be sort of comical. And look at these titles. Look at how like hoity-toity they are. He's the First Lord of the Treasury and the Lord Chief Justice and the Commander Lord High blah blah blah. And that, by the way, is the opposite of the people that I get to work with who do all these things, because none of them ever want any credit and none of them ever take any credit, and they're all doing it because it's the right thing to do, and they're not looking for these great titles. There is this other reference that you'll find if you try and figure out where this is from, to I guess somebody who was the chairman of ABC, and he was asked something in the Village Voice, and he asked me, he says, I wear 2 hats. Are you asking this question as president of the Bartenders Union or as the chairman of ABC? And so, out of context, it's kind of funny, but the point is that sometimes we wear different hats that might have some conflict or might have different interests in them. And, by the way, this is not the Grand Poobah. This is Sam Slagheap. He's the exalted Grand Poobah of the Loyal Order of Water Buffaloes from the Flintstones cartoon, which I think must have been in deference to Gilbert and Sullivan. But this is what I mean, right? This is that pipeline I showed you before, and then all that stuff down below there, these are slides that I borrowed from people at clinical research learning institutes or advocacy things. The ones that say ambassadors in action are all from Rick Bedlack. But this is kind of showing how people living with the disease and research warriors are impacting every phase of this. So, having these engagement sessions to help make the registry work better and help improve its catchment and its analysis and its data sharing, and helping, obviously fundraising, but then, what the ALS strategic planning at every stage of this pipeline has been impacted over the past, I would say, 2 years in particular, by people living with the disease and their families and their friends. And it even carries through to recent drug approvals and testifying in front of the FDA and really pushing different kinds of endpoints and study designs and making sure that studies are done in ways that are equitable. Some of you may have known Kathy Morris. She's passed away. She started this thing called the Morris ALS Principles, and the motto is nothing about us without us, right? And she gave one of the awards was actually given to the DoD program, the ALSRP that Matt and I volunteer with. And it says here that the ALS research program included people living with ALS on all scientific advisory bodies related to ALS research since its inception. That's included people with lived experiences of ALS as consumer reviewers of research grant awards, pretty much at every stage, right? And so these Morris principles are just that. It's that at every stage, whether it's conducting a clinical trial, designing a trial, reviewing research grants, that we really are sharing hats. We're really all doing this together. I'm not going to talk about the program, that program much today, but if you're curious, go to the website. Just Google ALSRP, and it'll come up. And there's a lot more information about the kind of work that it does. I do want to tell you a little bit about the history of it because it's relevant to the way that we all work together. So the ALS Research Program, originally, this is a DoD program. It's called Congressionally Directed Medical Research Programs. And it started in the 90s, and it started as a breast cancer research program. And it started because of grassroots advocacy. And even from the very beginning, they involved what they call consumers, which are people living with the disease and caregivers, in every aspect of program design, award design, review. The ALS part of this was added in 2007. Thousands of people living with the disease have served on review panels, have served on the programmatic panel at every stage of this disease. And this is why I love it so much, and I volunteer time to do this because, I work for you. So, how do we develop therapies though, right? Like, how do we fund research? And, this gets to a little bit of some Matt talked about too. What is ALS? How do we search for targets? Like, how do we decide what we're gonna test? And what does it mean when somebody says, oh, I have to validate these results? Like, what really does validate mean? We hear a lot nowadays about biomarkers. What does that mean? We're gonna talk about that a little bit and how we use them and why they're changing the way we do research. And this is important because, Matt, where is Matt? He ran away? Okay, so Dr. Harms, pardon me, talked about that a lot of trials fail, right? And we all know that, even though we know a lot of science. And so if you own a pharma company, do you want to invest in ALS? I mean, in order to have an investment in a disease, if you're going to pay all this money and run a trial, you don't want it to fail at the end. So we talk a lot about how can we de-risk these investments? How can we de-risk pharma companies' investments in ALS? And, I hear a lot of people say, well, why can't we do an ALS like they did in cancer or they do in HIV? And what does that mean? And why can't we? What's the difference? And importantly, at the bottom there, for the first time, we're learning how to harness this interconnected world we live in to really improve a lot of these aspects of therapy development. And so this is the main problem, right? So ALS is a variable disease. It looks different in every person. It can go fast, it can go slow, it can start in one place in your body, it can start in another place in your body. And we know a whole lot about the biology. It's amazing. I mean, Dr. Harms, pardon me, just gave a really remarkable talk about these genetic advances. The way I think of this, so I'm on service right now, and there'll be patients admitted to the hospital, not with ALS, but with another disease, and we don't always know what's wrong with them. But we have an evidence base that tells us if we see this on exam or this lab that this treatment's going to work, right? Even when we don't quite know the diagnosis. And the way that I often explain it to patients, and it made me think of it because it's Lou Gehrig's day this weekend too, is I say, we don't always know, we don't know the team that we're playing against, but we know how to play the game really well. And if we play the game the right way, people get better. And ALS is sort of the opposite of that for a long time. We know the color of the underwear that the other team is wearing. And yet we don't always have a play. And so, why is that? And why is it that we can know so much about the biology and things aren't translating, right? Translating from a mouse or cells into successful therapies. And so the questions we ask, are we using the right models? And one thing we've learned is that there's different flavors of ALS, right? And if I develop something in one model, maybe I have to figure out which people living with the disease should get that therapy. And if I'm targeting a particular pathway, Dr. Harms talked about different pathways that have been invoked, maybe that's something that's only going to work for some people. So how do I figure out who they are? And as you guys all know, the real issue is that it's not like, we don't have a test. If you don't have one of the genetic forms of ALS, it's not like I can, measure your blood sugar and say you have diabetes, or take your blood pressure and say, oh, you've got high blood pressure. ALS, the diagnosis relies on somebody like Dr. Harms or me examining you, and it's a clinical diagnosis. And it can, again, start anywhere in your body and spread anywhere in your body, and it goes at different speeds. And so because of that, it's very challenging. Like what's different about a tumor? Well, if I have a therapy in trial for a tumor, I give the medicine, and I can tell if the tumor shrunk or got bigger or didn't change. If I have somebody who's got HIV because they have an immune disease, I give them a drug, and I can see whether their immune system changes. And in ALS, it's much more challenging because you got to go to your doctor, and they got to say, oh, your weakness is a little more in this muscle than it was the last time I saw you. Dr. Harms talked about all the genes, right? And so this is a figure that's outdated now from 2015, but it shows a bunch of the different genes that have been discovered for ALS. And so this is years here on this axis. And the size of these circles is how common they are. So C9orf was discovered right around here, 2011, and it's the most common. And the second most common is that first gene, SOD1, right? And the number of genes keeps going up as far as the ones we're discovering. But those are still 10 or maybe 11% of all the cases. The rest of them are sporadic. This is a figure that Matt actually made for me for another talk that I gave a little while ago, and even this one's out of date now because geneticists like him just keep freaking discovering more genes because they keep finding new ways to do it. And the number of genes keeps going up and up and up. One thing, somebody asked a question, and I agree with them. One of the things that we still don't understand, like these genes do things in our cells that are very important to every cell in my body, right? And so we still don't understand what we call selective vulnerability. Like how is it that these different genes that are implicated in different diseases or do something in all of the different cells in my body, how is it that we end up with this very specific thing called ALS? I wanted to point out that first gene, SOD1, so it was discovered here in 1994. We had our first therapy, Rizal, in 1995. It wasn't tested. Oh, by the way, we had a mouse, right? That first mouse, the SOD mouse, was 1994, okay? Rizal, 1995. It wasn't tested on those mice, but can you imagine back then, they must have thought like, hey, we're gonna have this thing cured in like 2 years. We got our gene, we got our mouse, we're good to go. And so this is the kind of thing, like that's, I guess, me in the bottom with my head exploding, right? When you could start trying to look up and learn about the biology, it's a lot of different stuff. I mean, these are just some examples of different cell processes that are implicated in ALS. And it's a crazy list because, again, just like it's a lot of genes, it's a lot of different things. And a lot of these things every cell in my body needs, right? So this thing here, abnormal nucleocytoplasmic transport. You think like most of my cells have to get things from the nucleus to the cytoplasm for everything that they do. You know, mitochondrial dysfunction. You guys remember like mitochondria from, you know, the powerhouse of the cell, like every cell. So a lot of mechanisms, and a lot of the, you know, these experiments that we do in lab, maybe they're on cells from a patient or from a model or on mice, and we think there's different cell types that are involved. So where does all this come from? Where does this mess come from? And how do we find these things, and why do we implicate these things? And it has to do with the way that we do research. Okay, so the way you do research in the lab is you have something, you have your normal and you have your disease. You have, you know, cells from somebody with ALS and cells from a normal person, or a mouse with a mutation and a mouse that doesn't have it. And you're comparing them to each other, and you're looking for something that's different. And sometimes it's really obvious, like, you know, an apple and an orange, it's pretty obvious, right? They're different. But sometimes it's less obvious, like maybe you've got two different types of apples, and by looking at them, you can't tell, right? But if you grind them up and if you make applesauce and if you then, you know, analyze that applesauce, maybe one's got more sugar or something like that, or something's different about it. And this is literally the way we do experiments, right? We look at tissues, we analyze them, we look for things that are different, things that we can change. One thing we wonder about in ALS, as the disease progresses, say, you know, you lose motor neurons. If I look at motor neurons in somebody who's got ALS and compare them to normal, maybe the ones that are left are actually the ones that were sort of protected for some reason, right? Maybe the really sensitive ones have already died. You know, it's sort of like if we're looking at these apples, is it different if we look at the ones that are still on the tree or the ones that have fallen to the ground already because somebody's not watering the tree correctly? And so these are the sorts of things that we think about. And the fact is, if we're looking at cells, if cells are apples and you grind up cells, you get applesauce, our tissues are more like apple pie, right? Because if I look in your brain or your spinal cord, it's not all neurons, it's a bunch of different kinds of cell types. And if I grind up apple pie, I'm not just going to get the apples, I'm going to get all the other ingredients that I'm analyzing too. And if I look at something that's secreted into your blood or into your spinal fluid, that's kind of like this ice cream that's melting off the apple pie onto the plate, right? And so we have to be sure that what we're measuring is a valid measure and that it represents what's going on. But this is actually the way we search for targets, and then you look for what might be different and see whether you can change it. So this is an example, right? So this is a normal mouse, and these are mice, I take that back. Both of these pictures are mice that have that SOD mutation, that very first mutation. So these are ALS mice. These are all litter mates. These are all mice that are from the same litter, and the ones on the left were treated with placebo, with saline. The ones on the right were treated with a drug, and it actually doesn't matter what the drug is. But, you know, if you look at them moving around, you kind of might appreciate that maybe the ones on the right are moving more, right, in general. But if you actually look at it for a minute, you'd be like, hang on a minute, because there's one dude on the right, he's not really moving that much. And on the left there was one that was maybe moving a little better. And why are they different colors? And why are some bigger than others and smaller than others? And so this is a drug, this is a particular treatment that actually I'm involved with, but you can understand how you've got to design, when Dr. Scheffner was talking about designing the experiment right, you got to have the right number of animals, and you got to understand how variable something is, and you have to make sure that you're comparing, you know, apples to apples. So this is my house. I live out in the woods in like off the grid in northern Baltimore County. And it's an old stone house. We moved in 10 years ago. And the only reason we could afford it is because it was completely destroyed. Like everything was broken. There were 50 broken windows. Furnace didn't work. Like we had to replace everything. But we did. We fixed it up and we made it ours and we love it. And then that very first winter that we lived in that house was like a super cold winter in Baltimore. And we learned that this house, which is, by the way, made of stone, 2-foot thick stone, is like the draftiest house you've ever been in your life. And if the wind blows outside, the wind blows inside. Okay, that's literally how drafty it was. And if you didn't know what made a house drafty, if you just said, you know, this house is cold, I don't know why, I'm gonna compare this house, the disease house, to the one next door, which, you know, is a modern home, and see what's different. I'll tell you what, I'll bet you the first thing you notice is that we had a lot of ash in our fireplaces, because we burned a heck of a lot of fires that first year, okay? Now, does that mean if you remove the ash, you'll make it better, right? Does that mean that, and interestingly enough, a lot of things in neurodegenerative diseases may not be ash in the fireplace, but there's stuff that accumulates where it normally isn't there. And so Dr. Harms was talking about TDP-43. TDP-43, we do think we understand So this is this picture on the right here, on the top part. This is a cell, that's the nucleus in the middle, that's the TDP43. And in ALS, it goes out to the cytoplasm and gets stuck there, right? And so it's not doing what it's supposed to do in the nucleus, and it might be doing bad stuff in the cytoplasm, we're not sure. But a lot of times we have to think about that, is what we're seeing ash in the fireplace, or is it actually matter? Now, let's say you knew how fires worked, and you knew that's why it was there, and you said, okay, I need to burn more fires. You know what we learned that first year? If you burn more fires, you actually cool your house down, because it sucks in the draft from like everywhere else in the house. This is my, because everything's connected, right? This is my furnace, okay? We have an oil furnace. We really, literally off the grid. This is a picture from the middle of the night, that first year that we lived there. This is an empty oil tank. That thing's supposed to be up here, because we ran out of oil, whoops, and we were absolutely freezing, and it was a disaster. My furnace, if you compare it to the next door neighbor's furnace, it might be a great furnace, I have no idea, but I guarantee you it breaks down more. Even if it's normal, it breaks down more because it works a lot harder. I guarantee we burn a whole lot more oil than my neighbor. If you concluded from that, oh, you have to burn less oil, that's what's wrong with your house, that's not the answer either, right? So when we make these comparisons, we think about these sort of things. It is interesting though, because, so that bottom left corner, a lot of the pathways that are implicated in ALS are kind of related to energy, right? Are kind of related to energy or energy transport. And, is it the furnace? Is it this kind of thing? Is it accommodating for the primary problem? And so, my silly metaphor, what's the point I'm trying to make? So ALS is a clinical diagnosis. We know there's a lot of different things that we say can cause it. And in a given person living with ALS, the disease is probably the product of different pathways. And in some people, some of them may be important than in other people. And so one of the things that we're doing better than we used to do is figuring out who those subsets are. Like, can we identify which people living with disease have a problem with the furnace, or a problem with the oil, or whatever it might be, or the windows? Because ideally, you'd like to address that underlying abnormality. I'll say that there is this, we still don't understand how all these different things can cause this very specific disease. And oh, by the way, not just this disease, the same pathways and genes are implicated in other diseases, right? Sleep apnea, Alzheimer's disease, prostate cancer, you name it. So how does that work? How does it happen that different general cell pathways can do these different things? So in my house, what's the genetic mutation? We've got steel sash windows from the 1940s that are single pane, that are drafty, right? Gene therapy, up until a few years ago, is hard. We're getting better at it. I'll tell you, we can't replace these windows. It's still too hard because the house is made of stone and there's not an easy way to do it. This is my father-in-law. He passed away. They would come visit us in the holidays and they'd like turn the heat up in every room, and we would spend like $90 a day in oil. And we'd follow him from room to room and turn the heat down. And so for Christmas, we bought him these footy pajamas, right? Now, this treatment of him had a great side effect, which is that it made him look ridiculous, which we all love. But it's actually an effective treatment because he didn't feel cold. And you don't necessarily have to address the underlying mechanism to have an effective treatment. And in fact, those mice that I was showing you is an example of that. So the drug that those mice got is this thing called MG53. It's a protein that makes muscle fibers be able to repair themselves better, right? So it's not targeting anything that's necessarily a problem in ALS, or may not be. It's making the cells be able to repair themselves quicker. It's sort of like wearing footy pajamas. I want to point out a couple things about this figure because it's the way that we see data sometimes from ALS. So see these tracings here? These are those mice walking around, the treated ones. And this is like, the mice aren't coming into my clinic and me saying, oh, how strong are you? Let me tap on your reflexes, okay? Some automated system is tracking how much they move around. And an automated system, wouldn't it be cool if we could do that in people, if we could, like an automated way, track how they're moving around and not have to just bring them into clinic? And maybe we can, and I'll get to that in a sec. What these other figures are showing is the kind of information that we'll see in trials. So this plot here, this is how many mice are living, and this is how long they're living. And this is showing that with the medicine, they lived longer than without the medicine. And this up here, this is showing that there's something we can measure in their blood, that by measuring it in their blood, we can, A, track how the drug that we're giving is staying around, but also we can measure response, right? And so this is the kind of thing that this is in a mouse, but if we could measure that in people, we could tell maybe, what was going on. Let me just, by the way, say, I'm not advocating for this therapy. It's one example of a zillion. I hope it works, but I hope they all work. So why do trials fail, right? How can we do all this cool science and then we bring something and they don't work? And I want to use a specific example, and the example was ceftriaxone, which is now several years ago, but it was the first trial, at least that I remember, where there was all this big hope that this was going to be the one that we finally had a treatment. And it came out of this, one of these pathways, that was this, it's called glutamate excitotoxicity, this idea that there's a transporter that's decreased in ALS, and it's on astrocytes, and if we increase that transporter, we could treat the disease. And so they screened a whole bunch of different drugs, and this was, Dr. Rothstein and colleagues at Hopkins, and they found that that ceftriaxone, the antibiotic, was something that was able to increase this transporter, right? This was one mechanism. And in the mice, so this is those same mice, and it's the same thing. So how many are surviving over how long? And if you give them the drug, they lived a little bit longer, okay? And it got taken to clinical trials in people living with the disease, and in phase 2 trials, it worked. We hear this a lot, right? And then in a phase 3 trial, it failed. It failed so miserably that the trial was stopped early. And when it failed, we didn't know why it failed, right? So what are the possibilities? Did it fail because it was just a dumb idea? Like, was it just not true? Is it something that maybe was true in those mice and not in people? Or maybe it's only true for a subset of people? Maybe we need to know which people living with the disease to do it in. Maybe it literally didn't get where it was supposed to go, right? Maybe if you give this drug, it was given intravenously, maybe it didn't get to the cells that it needed to have this effect, or maybe it didn't have that effect. And we didn't have a way to know those things. So when this trial fails, we don't actually know what the problem was. Maybe it would have worked. Maybe if we had a way to tell, if we had a way to measure those levels, we could have answered that question. And so this is where biomarkers come in, right? And this is an old press release from ALSA. I can't see my eyes. It's like 2014, I think. Yeah, so, a biomarker is something that tracks the disease process. This is back in 2014. We've gotten a lot more savvy about this now. This is an oversimplification. And why do we want biomarkers? Because pharma companies don't want to do trials that fail. And so we need to de-risk trials. We need to have a way to measure something that lets us know whether our drug is doing what it's supposed to do, whether it's reached its target. As we've gotten better about this, there's actually different kinds of biomarkers, right? And so these figures are thanks to Dr. Benatar, who's at Miami, who runs something called the CREATE Consortium and does a lot of biomarker work. So this is one kind of biomarker. It's something that you might measure one time, and it's one level in somebody who doesn't have a disease and one level in somebody who does, right? And so this is what we call a diagnostic biomarker. And a gene test would be a diagnostic biomarker. Either you have the mutation or you don't. And well, actually, a gene test would be, I take that back. A gene test we would call a predictive biomarker. So if you want to go into a therapy for, tofersen, you take the gene test, and if you have a mutation, that means maybe that drug will work for you, and so the test is predicting whether you would respond to the therapy. If you want to go into a trial that's not a gene therapy, if you want to go into a trial for something that makes mitochondria work better, we need a test to tell that you're somebody whose mitochondria aren't working well. We don't have that, right? We have that for the genetic forms of ALS, but not really for the non-genetic ones right now. So this is an important kind of biomarker. So this is something that you measure one time, and then if it's high or low, this is survival over time, and this is maybe getting weaker over time. If you have a high level, maybe you're going to survive longer. If you have a lower level, maybe you're not going to survive or you're going to get weaker. And so what would you call this? So this is a prognostic biomarker. This tells us what your prognosis is, right? And this is important because some ALS patients progress quick, some progress slow. And if you have a clinical trial, you need your control group to look like your other group, and you need to be sensitive. And then this is sort of the most important. So each of these tracings is a different person, okay? And so it's something that we're measuring, let's say, in the blood or spinal fluid, and maybe it stays the same over time in somebody with ALS, or maybe it goes up over time. But then you give your drug, and you can see it go down. Or you give your drug, and you can see it stop going up. And this is called a pharmacodynamic biomarker, and this is what we really need, right? Because we need to know that the drugs are doing what we think they're doing to know whether they're working. And the fact is, we actually have a lot of candidates in ALS for lots of these different biomarkers. But we need to do certain things to get them clinical trial ready to be able to use them. And so this is one example. You probably hear about this one a lot, neurofilament light. And this was a study that Dr. Benatar and the CREATE Consortium and I sort of worked on together. And what we had to do was we found every commercial company that analyzes this one thing, neurofilament light. And we sent them all the exact same samples, and for every assay that they analyze it with. And we tried to see how did they perform against each other, right? Because if you're going to do a trial, you need to make sure everybody's testing the thing the same way and it's reliable, and if you test it over and over, you get the same response. And so this was a way to validate which assay actually works and which one is going to work in different people's hands, in different companies' hands. This was part of the data from that trial. This is survival, and this is time, and again, you measure one time, and this looks like that other figure that I just showed you a minute ago, right here, right? So this is a prognostic biomarker, and as you might know, we think that NFL is a prognostic biomarker, so we think high or low levels tell us if someone's, this might be Dr. Patterson, hold on. Okay. Now, these are all things that we measure in tissues. Remember those mice that were walking around in the cage, right, and we were tracking their measurement? So there's a lot of interest right now in what are called digital biomarkers. And so the one program that I'm involved with is Everything ALS, but there's also ALS TDI, there's several other efforts that are doing similar things. And the idea is, if there's something that we can measure about someone at their home, doesn't require coming into a brick and mortar clinic, doesn't require living near a clinic, doesn't matter if you have good insurance, measure with a cell phone, measure with some sort of digital technology, and maybe measure more frequently too, right? Because Dr. Harms and I, we might only see patients every 3 months. But what if there was something you could do in your house on your phone, or not even on your phone, automatically, that was collecting data like those mice walking around? And so this one study, this is called the Radcliffe study, it's actually going on right now. It does a couple different things. It's smart shoe soles that go on your shoes that have all these different sensors on them, and they're Bluetooth. And we send you a phone, and we send you the shoe soles. You put them in your shoes, and there's walking tasks and stair climbing tasks that you do as part of this protocol, and all of that data is automatically collected. This study also has a Bluetooth-enabled breathing tester, a spirometer. And then it's got a phone app, and the phone app analyzes speech and analyzes facial movement. And the idea is that by analyzing these different things and having artificial intelligence and machine learning, that hopefully we're going to have markers that are much more sensitive than we have now, that can be done without bringing somebody to clinic. And by the way, this was like 2 days ago. Somebody sent me this picture. Apparently, I'm in the middle center there, and I don't remember who sent it to me, but my wife and parents, I think, were more proud of this than my actual work, because when you get your name on the lights in Broadway, anyway, something like that. But this is the thing, right? So compare that to what we have now. So if I'm analyzing your speech from an app on a phone, listening to you read something, having a computer analyze it, what we have now is a 4-point scale when you go in to be in your clinical trial, this speech part of the ALS FRS. You either got normal speech, your speech is a little not normal, your speech is really hard to understand unless you repeat, or I can't understand you, right? How are we going to detect subtle changes to know if a drug is working to help somebody's speech with that? But that's what we have. And as an example, so I'm going to play this recording here. This is somebody who has scored themselves as normal. So it's too quiet to hear. Can you turn up the volume? Anyway, we'll get the volume turned up for the next ones, but I think you can easily appreciate that speech is not normal, right? And so to have such an insensitive scale. Perfect, that's good. And so if we do these speaking tests and then we let the computers analyze different things about them, how fast is someone speaking? Like, I speak way too fast, I'm sure you guys notice that, blah, blah, blah. But different people speak at different rates. Different people have different intelligibility to their speech. These algorithms that analyze all this different stuff, and they can figure out clusters of different kinds of people who are progressing at different rates, people who are following different patterns. You can combine the speech part with the walking part, with the breathing part, and the idea is that we're going to have something that's going to be able to, much more sensitive, tell us when somebody's changing. And what we're finding is we're doing this sort of thing is that different stuff changes differently, in different people. So this is a plot that doesn't matter, looking at speaking rate and looking at how easily this computer can transcribe them. And I just want to play for you here. So both of these recordings, again, were people whose speech was rated 4. So this is normal speech on ALS FRSR. It didn't play.
They provide a good background and create the mood in Japanese gardens.
So, believe it or not, that one, the computer's able to understand with a decent confidence. It's hard to hear in this room. This one, the computer's not able to understand, but. Yeah, it's not playing so well for you guys. But in any case, I think you can appreciate how these sorts of tools can be much more sensitive, right? And not just more sensitive. Dr. Harms was talking about how all this work has been done in white males, and that's true, right? These sorts of tools, you don't have to live near a clinic. You don't have to, you can be, when we talk about care equity, when we talk about reaching underserved populations and people who, maybe don't live near a big center or something like that, anybody can have a phone app, and anybody can participate in trials and have this stuff tracked over time, and you can do it much more frequently too. And so this picture's here. So this is Dr. Hyman Patterson, who's covering the inpatient on rounds for me right now because she's like infinitely energy. This was her a couple weeks ago out in the rain at a Latino health fair promoting sort of care equity and care outreach. This was the day before the ALS Hope Foundation award ceremony, which is here, which, by the way, Dr. Harms was the speaker at this year. And the reason I wanted to show this slide is that there's this group End the Legacy. So one of the things that Dr. Harms talked about, all the genetic testing, the more and more genetic testing we're doing means that we're finding more and more family members who don't have a disease but carry the gene, right? And so people who don't yet manifest the disease, and we have to figure out how to do trials in those people. We have to figure out what should we be monitoring? How frequently should we be monitoring it? When should we give them medicines? How do we know if the medicine's doing the right thing? And I have to tell you, I think digital biomarkers could be wonderful for that, because if I give somebody a Wi-Fi squeezy bubble or something in their hand and they carry it around with them and they just squeeze it every day, I'm going to get data on them and they're not doing anything, and it's, there's measures, or like those mice walking around. If I have smart shoe soles that are just watching somebody walk, maybe we're going to be able to tell when somebody starts changing. And so I think there's a lot of potential there for those sorts of people. And the Legacy is a group that some people who are from families that have these diseases, look them up. They're remarkable. They are just moving the needle like never before, and it is so amazing to watch and so inspiring. And some of you in the room might be friends with some of them. But in any case, this is something that I think we're going to see more and more. When do you treat somebody with a genetic mutation, and how do we figure it out? So Nadia, I mentioned Nadia is on our programmatic panel. Every year when we do our funding cycle for the Department of Defense, we do analyzing the landscape, figuring out what we should do. And I'm not going to go into the details of this slide, but while in the past 2 years, neurofilament has gone crazy and is used in a lot of therapy, a lot of trials, we still don't have mechanism-specific biomarkers. If somebody's doing a trial of a complement inhibitor or a growth factor, we still don't have good ways to tell which people living with the disease should be getting that therapy, and it's an unmet need that we're trying to address. Joe Lukac is from Denali. He's also on the panel, so we have industry also sharing hats with us and trying to help us figure out those exact things. This is an interview that you can read that's on that website. And this is why also having biological samples from people living with the disease are so important. Blood, spinal fluid, tissues after they pass away. I run a post-mortem research corps which collects autopsy tissues and data and makes that available to researchers around the world. And the reason is because we need to find measures that we can measure during life, that we can tell which people living with the disease have which of these things going wrong. And in fact, that's my involvement with those mice that I showed you. So this picture on the right, so those were experiments in mice done by those other researchers. These pictures on the right are fluorescent stained muscle, diaphragm muscle, breathing muscle. And some of them are from people without ALS, some of them are people who have a non-genetic form of ALS, some have a genetic form of ALS. And I think what you can see from this picture, see how smooth this looks? And see how like lumpy bumpy this is, right? And the lumpy bumpy's in the genetic ones, the non-genetic ones, it's not there in the normal muscle. It's exactly like in the mice. So this is the mouse's normal muscle and the ones that have the mutation. So this is a way that we can say, look, we see the same thing in the muscle of people that, that we do in the mice, that we can validate it. So the last thing I just want to talk about is how do we fund research, right? Because we get asked a lot, why aren't you guys doing this? Why is this not being done? And how does that happen? So, so this idea about funding cycles and grants. So the very first NIH study section met in 1946, okay? And it was a study section about syphilis. And this was the very beginning of what's called peer review, where people are reviewing grants that aren't part of some, good old boys network, but actually are experts and scientists. And this peer review process is what helped the U.S. really lead the world for many years in medical research, and has been the model that even like MDA or also everybody has peer review proposal cycles, and really adopted by everybody. But the thing is, that process has sort of remained unchanged since 1946. And so the problem with it is this idea of a funding cycle. And what happens is, an organization decides they want to fund something, and so they write a request for applications, a program announcement. And then researchers reply to that. They write a proposal. And then there's a round of review of those proposals. Sometimes there's even a pre-round of review. And all that takes a whole lot of time. And then everything gets ranked. And then they have some meeting of an executive council that says, oh, here's what we're going to fund. And again, this is this long, drawn-out process. And because of that, only a small percentage can be funded, right? And so a lot of really cool stuff doesn't get funded. And a lot of really great ideas. And even in the best case scenario, it still takes many, many months to get a new idea funded. And reviewers, when they're reviewing these things, they're considering, like, is this well designed? Is it well powered? Are there enough mice? But they're also considering, is this the right target? Is this the right disease? And I'll tell you what, I don't buy that anymore because, like, supposedly Dr. Harms and I, like, we're ALS experts, right? What does that actually mean if I can't treat my patients, okay? It means that I'm an expert in, like, the multidisciplinary care, and I'm an expert in understanding how to diagnose the disease and see its tracking. But if a scientist tells you that one particular pathway is the answer to ALS, look, don't believe them. Now, that doesn't mean it's not the answer. It means we have to test it. It means we have to have rigorous ways that we test these things efficiently, and we've gotten a lot better at that. I just wanted to highlight, so back then, in 1946, study sections had two jobs. They weren't just reviewing grants. Their other thing that they were supposed to do was survey the landscape of everything going on in an area. What's the exciting research? What should we be funding, right? What kinds of research is out there that we should be interested in? This is that first study section on syphilis. Really diverse, right? Do you see people living with the disease in the room? Do you see women? Do you see anything except white men? Like, the only diversity in that room is that some of them have dark suits and some have light suits. Okay, other than that, oh, and then this is the other thing. So this guy was the chair. He's actually from Hopkins. I don't remember his name. But he was the person who discovered that penicillin works for syphilis. And he's chairing the committee that's, like, giving out grants for other causes of other treatments for syphilis. Anyway, I did notice in this picture there's some cool hats in the back. So maybe they wore different hats too. But we've come a long way from this, right? And because we do things differently now, we've designed trials better. We have ways that we're all working together. And I generally talk for too long. People who know me, sorry, but almost done. This is what the cycle looks like for the DoD. And so Dr. Harms, me, and I both serve on this programmatic panel. We're involved. We do that same thing. We do this. We every year, members of the programmatic panel do a whole bunch of research, and we go out and find the expertise that we need to say what are the important things that we should be doing. And then we, release a program announcement. People write pre-apps. Those get reviewed. Those then go out for the ones that get invited to write a full application go out for peer review. People living with ALS are involved in every stage of this process equally and fully. Those then get peer reviewed. They get ranked. They come back. There's something called programmatic review. This is what the cycle looks like. These are the dates right now, if you're curious. So we're right here, right? So we've done the pre-app review, and we're about to get these applications. This is what the funding for that program looks like, and I joined this programmatic panel here. And this is the number of awards that could be given each year. And we would get 150 applications every year, and I'll tell you what, even if you do the best job possible, who cares? It's ridiculous. You fund 3 awards, right? It is completely because of advocacy of groups like MDA and ALSA and IMALS and ALS Research ambassadors and people living with the disease that this happened, right? And I actually became the chair here. This is not because of me, this is because of you. I just got to have the fun of helping figure out and being responsible and making sure that we're using the medicine the right way. There's no other program that I'm aware of in science that has a funding rate this high. It also meant, now that we have this larger budget, that we were able to move into funding clinical trials, right? Because clinical trials are expensive, even early small ones. They cost a couple $1 million each. And we didn't want to jeopardize the work that we'd always been doing in finding new, innovative, novel therapies, which is what our program's kind of known for. But once we had those funds, we've moved into clinical trials. And we've also moved specifically into grants to find biomarkers. They can be biomarkers in your blood, in your spinal fluid, digital markers, telemarkers, things, and the biomarkers we really are emphasizing now, based on our review of what's going on in the world, finding ones that are mechanism specific, okay? So again, things that might tell us which people are going to respond to a specific drug and how we know in the trial if they're responding. The other thing that we've done is that for all of these mechanisms, So these are these are like early ideas. This is, you know, get something to a clinical trial. This is do the clinical trial. Biomarkers have to be incorporated in all of these. If the researchers don't already have them, the proposal has to develop them. You know what else has to be incorporated is data and resource sharing. And people made a big deal last year when NIH said that everybody has to, I don't know if you guys know this, people who write proposals to NIH are required to write a data sharing statement, but they're just required to write it. It's not evaluated, it's not scored. We actually score it, okay? It's part of the evaluation. If somebody just phones it in and doesn't actually have a good plan, that counts against them. This year, for the first time, not only are we doing clinical trials, but it is a requirement. We will not fund a proposal for a clinical trial if there is not involvement of a community partner, people living with the disease, a patient advocacy group, an advisory council, and not just involved in every step, trial design, trial execution, trial evaluation. It's a requirement. And I got to tell you, when I think about what's changing and why we're being successful and why things are finally showing a signal, it's that, okay? It's the fact that we're all doing this together, that we're not a pipeline, that we really, literally are working together at every phase. And we're learning how to use things like technology and digital markers. And I'm going to skip this because I've talked about too many other things already. This is my kid and my wife. My son just finished his freshman year of college. This was when we were dropping him off at the dorm, okay? At the first day of school, that's his dorm room. And while we were doing this, you know, I was looking back through some old pictures. So this is me like 35 years ago, my first day of college. And I noticed something when I was looking at these pictures. I noticed two things. One of them is, why did I have a weird hat in my dorm room? I don't think I actually wore it, but anyway. But I noticed this thing on the wall in this old picture. And the reason I noticed it is because I'm not somebody who hangs like inspirational statements on the walls, but that thing is still on my wall. And I just moved from Johns Hopkins to Temple. And I moved to Temple to be with the ALS Hope Foundation and the MDA ALS Center of Hope, and Sarah and Mark, who you're going to hear from, and literally because I want to take care of my patients the way that they do. And this statement is how I think about every ALS warrior that I know, every person living with the disease, every advocate, every caregiver, and it inspires me. And it's just remarkable when I look and notice that I had that same thing hanging over my freshman dorm room. Sarah and Mark are going to talk to you about you know, some really remarkable stuff that I know nothing about in a bit about how to care for people. These are newsletters that Sarah writes every month. That it is just remarkable how much is going on and what an exciting time it is. And drugs are getting approved, and all this progress and all of these events are because of you. And I work for you, and Dr. Harms works for you, and it's really not the way it was even 5 years ago, and you know, it's why I think we're making some progress. So thank you.